Mark Well. My project consists in developing a gene editing analysis pipeline in a Spark R. So basically, the aim is to implement a computational method to analyze gene editing experiment results. So basically, very briefly, a gene editing experiment consists on using a vector or a genetic tool to modify a certain loci of the genome of a population of cells. Then, to measure how this experiment was performed, what were the results, basically what we do, we amplify these regions that we were targeting, and then we deep sequence this data so that we can see how efficient was our editing. So I decided to implement this in a Spark R because I think it's a very good synergy between R, which has probably the widest variety of genomics software libraries, and the very high capacity for large data of a Spark. So in this case, the input data is a large text file which comes from the sequencer that contains all the sequences. And then what we need to do is to compare all the sequences to the reference sequence to basically to measure and analyze how much and which changes we induce to the population of cells. And for that, we use a Spark. So this is how, what is the output of the data, how I represent it. So basically, first, I am showing here what is the percentage of changes. Here, where the changes are present in the respect to the reference. Here, the deletions, and here, the insertions. Then in these two plots, I'm showing how large are the insertions or deletions. In this case, deletions, and in this case, insertions. Then here, underneath, I'm showing a tool to browse each individual read. So we can go zoom out, zoom in. We can go into each individual read and see the different properties. So yeah, so that was the summary. Thank you.